Thank you very much. So I think, like Julia said, there's some of my slides here will be familiar to you. A couple of them I did show in the talk yesterday, so they are there as reference points to come back to. Strategic investment. So, of course, it's good to run assays, it's good to have data. Like the models which I was talking about, some data is more useful than others. So if you can make a decision from that data, that's obviously a lot better than in terms of investment than just having the data to hand. So again, kind of thinking if you did every assay on every program, I mean, it may be affordable, it may be the data you need, but there may also be decisions you can take where you don't necessarily need to gather all of the data on all of the assays. So I'll try and take you through that. So just kind of thinking about the cost of immunogenicity for programs, and a bit like Julia was describing, this is the bad cost. This is when things go wrong. I've got a couple of examples in here, some of which you'll have heard about, and I want to go into a little bit more detail on. Just a reminder as well about the IRA. You've been hearing about this risk assessment. This is a tool that will aid you at any part of the project, so whether that's a regulatory submission, but actually more often than not, we're using these to make these project decisions, these investment decisions. When is the right time? What stage of my molecule development, what kinds of molecules do I have to actually make a decision on? And therefore, does it make sense for me to do an assay in a standard way or sometimes in a non-standard way as well? And certainly our friends at ProImmune will be in a good position to help you work out exactly what to do with that. And then I will go through again some of the applications of the tools, really trying to highlight potential differences in the tools and how they can be used. And probably here to try and reiterate, there is roughly an order in which you would apply them, but you should see that as kind of a strategic layout rather than a map you have to follow and do them in that order, if at all indeed. So the cost of immunogenicity to programs. You've heard a little bit about this anti-PCSK9 antibody called boxizumab. So in part because this was in cardiovascular disease and the outcomes that they were trying to measure basically took a year and needed a lot of subjects. I think these studies actually cover probably about 9,000 patients, so really seriously big studies. So headline figure was that immunogenicity contributed to the termination with phase 3 data in hand. And that's kind of on the top right here with the 15% of patients who lost efficacy. So there's a lot of context to this. At the time, there were 2 other drugs which had very similar PD effects. So here we see the LDL cholesterol dropping nicely, staying low in the ADA negative and indeed the low titer ADA populations. And this was the case for the competitor drugs. So it was literally these 2 groups here which accounted for this 15% of patients, the higher ADA titers, where there was a loss of efficacy. So it wouldn't necessarily make sense for a physician to prescribe this drug compared with the other 2 drugs where there wasn't that chance, or certainly not that large a chance of losing efficacy. So there's some other things in this figure as well which are quite helpful to look at, and this is the incidence of ADA. And we talk a bit about incidence and prevalence. So incidence would be the ADA in the population over the whole time period studied, and the prevalence is the ADA measured at the particular time point. So the sort of darker bar here is the one as it increases over time, up to 48% of the population receiving the drug had generated an ADA response. And then at any given time, you can see it sort of peaks in the middle of the study and starts to drop off again. So this is also fairly typical of the profiles we see for clinical immunogenicity, that there does seem to be I'm cautious, I don't want to say tolerance because we don't know it's kind of an active tolerance, but sometimes the ADA are transient. So it's really important to understand, what's going on in these patients, and the likelihood is that those patients either just stay in this lower ADA group and rather than going into the higher groups. So again, when you've got large data sets, you can assess the impact on the population and again begin to potentially model the impact over time. So in this study as well, there was I would say a strong correlation again here with the drug level itself. So as they came to the end of the study, obviously the drug levels were going down, even in the ADA negative and the low titer ADA. And then the higher titers, it's pretty much matched the effect that we would expect with regard to the PD. So sometimes we talk about the impacts of ADA, one being safety, which is very important. There were also injection site reactions with this drug. But then also we talk about the PK, the PD, and the efficacy. So they don't always go together. In this particular case, they did go together. So loss of exposure, loss of PD, loss of efficacy, it was all together. So the other thing I wanted to highlight, I've already got the red box on, is with a lot of these kind of drugs, I'd say relatively, say normal monoclonal antibodies, so nothing too fancy. In those early clinical studies, you may only get up to about 12 weeks worth of data anyway, and you couldn't sort of judge that early on that was going to become a problem, and therefore, the longer clinical trials certainly do help, and it is our aspiration at present to be predicting for the impact of the ADA, not just the incidence. So depending on how you integrate the data, we certainly are improving at predicting incidence, and then the mechanisms that can lead to clearance are starting to be understood as well. And the other issue, of course, not necessarily just clearance, but potentially neutralizing capability. And there's some nice publications highlighting relationship between the ADA affinity and indeed off rate and neutralizing activity. So there's a lot to take into account. This paper is worth looking at as well because it does show nicely how you can present any ADA data on your clinical programs for some clarity. I realise it's quite complex in the top figure. It's worth working through this to help you explain the clinical data you see and to understand the data as it gets published in more standardised formats now. Okay, so just I wanted to highlight quickly another example. This was a Novartis drug, brolucizumab. So this is for treatment of retinal vasculitis. So the idea here is that you put the drug in and it calms the inflammation in the eye, and people who were starting to lose vision, in some cases may have that restored, but certainly no further loss of vision. So with this particular drug, it was launched, and then I think it was about within a year of launch, there was actually quite a few patients who were starting to lose vision more rapidly. So there was the investigation that Novartis did. There's a couple of nice papers from 2023 looking at ADA as being the cause, but also then why was the ADA occurring. This is one where there was quite a bit of pre-existing ADA, but also they identified this sequence which looks remarkably similar to a bacterial sequence. So that whilst that may have been part of the pre-ADA issue, this also contributed to treatment emergent ADA, which then led to the safety signals. So this is a drug that's still on the market, and I probably don't have the correct numbers to hand, but suffice to say that this is making a lot fewer sales than anticipated. And it's probably lost 90% or more of the anticipated market because of this safety issue. So the risk assessment to aid decision making. So we've heard about the IRA, you've seen this cartoon a time or two. So just a reminder that we do understand a lot of the aspects of the immune system. We're always looking to dig a bit deeper, and particularly if there's something around mechanism of action of our drug that we might need to understand, that's where we would go. But again, this is a fairly typical, let's say it's an oversimplification of the immune system to immunologists, but quite helpful for trying to explain sort of a normal immune response as we get with our drugs. This I showed yesterday as well, and speaks a little bit to the FDA guidance. I know we had the question before about route of administration, so it is there in the guidance, and you are expected to mention it. And I think, as well as Julia just said, the real key is if you start early, you get a chance to influence all of these product-specific factors. If you're picking up the chosen candidate, there may be some things you can do about some aspects of, the manufacturing process that could influence these, but you're set with a sequence, and there may already be things that are you're stuck with for when you go into the clinic. Okay. I wanted to highlight here, there was the European Immunogenicity Platform, EIP, has got a number of papers out recently. So this one's actually on the IRA. I would still say it comes from the point of view of a development risk assessment, and you can look at all of these things a bit earlier than they would state in there that you should look at them. And just to highlight, it's the same things you've seen elsewhere, but just with the paper kind of covers some examples of how you might set a particular risk level. I think the other piece of this would be, at what point, I'll explain. So it's traffic light basically, right? So if you're green, you're low. If it's red, it's high. The yellow's somewhere in between. And then I think there's quite a lot of discussion about having these sort of half and half. Is it okay to have low medium as an outcome? The other challenging thing is how you add all of these pieces together. So we've heard integrated quite a lot. What we don't know as a community, if you like, we don't know precisely how to integrate. So do we add these up? Do we take an average of the factors? It probably does take a little bit more of a mindfulness about not only whether you're getting the ADA, but then what the consequences are. So I would always advise people to think about which of these factors are contributing to us having the ADA and which are the consequences. And as they highlight here as well, this kind of cross-reactivity to endogenous counterpart with unique function is always going to be a flag. It's always a big risk. It doesn't mean that everything's lost. You just have to work harder on ensuring you're less likely to get the ADA, and hence again, more important to look at designing quality in the first place. Okay, so the application of the tools. I did show this one yesterday as well. So to highlight that we're not expecting to predict immunogenicity from the assays we use, because the assays, we're generally looking at the product and the molecule and the sequences and if it's of human origin. And then we might have to think about, are we really looking at impurities? Are we testing different levels of impurities in our product? What's going on with the pharmacology? Do we even have the target present in the assay and see, say, if it's a multimeric target, are we taking that into account in our assessment? So to get, so with the standard assay, we cover kind of the dark blue. And then we have to look at the specifics of the assay to try and answer some of these other questions in the lighter blue. And the grey we're not really addressing in these in vitro assays. We've heard a bit about the patients and the genetics, and Zubin Sorna's algorithms to pick the right patients is quite helpful. Sometimes you don't need to predict, you don't need a big HLA spread, because you might not be actually trying to predict immunogenicity for a cohort. You might be saying, I've seen a signal in a MAPS assay, and I just want to now look in those donors that can present that peptide and see whether there's a T-cell response. So you might then skew your population towards donors who are going to present. Word of caution on that, if you do that, make it very clear to the teams you're working with when you get an 80% score back, that's not 80% incidence that you're going to get in the clinic. So just a bit of caution. So I want to talk about in silico as well, because it can feel like a bit of a black box sometimes. I know I'm frequently asked for a number from the in silico, and it's like, well, what number are you looking for? Again, ADA incidence isn't going to be predicted by in silico T-cell epitope prediction, but there are a few things that you can do. Like if you're applying this very early on, say you've done a phage display, you can use a scoring matrix to then apply a cutoff and say, you know, we've got, I don't know, 50 hits here, and we can move on the worst 25 of them and then concentrate on other aspects of developability for the further selection. Sometimes a global score can help if you've already got an idea of, you know, how many epitopes, what's the potential strength of them, and you're looking to optimize. For sure, you want to reduce that score, so you know you don't necessarily get to, as Julia showed, the zero epitope scenario. You're probably going through rounds of how do we reduce the risk, but that can be done by looking at the score. The really important stuff is then when you can say, here's an epitope. You know, our protein engineers can see that there's you know, a sequence that doesn't interact with a target, or they don't think it'll interact with a target. Molecule will still be stable without it, so you can then change that. Always seems favourable to turn them to germline for antibodies where possible. There may be cases where it's worth doing something differently than that, but as a rule of thumb, combine this with the germlining. It is helpful to have higher percent of human content, but again, it's not the only metric you should be looking at. So you can kind of look at this average epitope content per sequence. When you look across the big broad data sets of marketed molecules, all of them have some predicted epitopes in. So a predicted epitope is not necessarily the end and a terrible place to be, but the transition from what's predicted to ultimately what becomes an epitope is something that we still need to further understand, and really the in vitro assays are the better way of getting to that. So one way then about thinking about this optimization as well is you can either sort of remove things so you're positively selecting these low risk sequences, or you can be a bit more active in engineering or derisking. So this is one of Epivax's Epimatrix from I think the old version, so it may be more over predictive than the newer version. But what we were doing at Pfizer was to kind of take some of these global scores, then look at the epitopes, and particularly these non-germline. I mean, they tended to be CDR, sometimes they'd be elsewhere. And then look at what's the likelihood we can optimize these. So each line is a different antibody, and there'd be ones where you go, well, you know, the global scores aren't great, the yellow ones, and we're unlikely to be able to optimize these. So we can get rid of these two at the bottom, put the other 5 or 6 through, and we're already in a better place. So this is even before you going into optimization or your lead optimization phase. So it is worth digging a bit into the details, because it's not necessarily that the global score gives you all the answers. So thinking again, I probably already said this, I want to emphasize it. So this looks like a nice pipeline, right? And you should follow this and do it on every program. It's not necessarily the case. Practically speaking, doing the in silico and finding these peptides, and we can use the ProMap assay here and get those peptides, the list sent off to ProImmune. As we were discussing yesterday, if you need them really quickly, you might have to have a conversation about which peptides you don't mind missing out if they're going to be difficult to synthesize. So you can plan ahead and know you're going to get data in time. So I quite like putting together the PBMC and the MAPS data. So if you've got your MAPS data and you know which sequences are presented, and you've already got that from a ProMap assay, then you're in a reasonable position to see where are the liabilities, what's going to cause me a problem, either when I put it into the clinic or when I put it into a more complex assay. Also, this combination's a lot more helpful to engineering teams where they can say, what was the problem? Let's see if we can do something about it. Sometimes if you only have the whole protein T-cell assay, you're guessing a bit as to what went wrong. Can we go back and do something about that? And by this time as well, you're probably wanting to put fairly clean, well-developed process material in. But through this process, so with your risk assessment in hand, you'll have chosen the right assays, you'll have driven to as low epitope content as possible. So you should be able to select for low-risk molecules and deselect the ones where you get the positive signals. I just wanted to highlight, this was a ProMap study. I can't quite remember what date we did this. I think this might have been 2016 or something like that. So back in the day when everyone was, you can't predict immunogenicity, the tools are rubbish, there's no point in doing them. We actually, with in silico and ProMap together, we played around with the odd sequence, and I've used the term de-immunize here, where we had some parental sequence that was pretty horrible. I mean, 49% in the ProMap assay is pretty horrible for sure. And there were variants that we knew we could drive to lower level. So then when we were looking at our other programs, we were in a position to say, well, we've got, I can't remember at the time how well established ProPresent was, but we were kind of on this one. Generally, it looked quite low risk, so we did the in silico alongside the other optimization steps, and we ran the peptides related to all of the CDRs, and then all of the frameworks that were also in silico hits, just to kind of see how that would come out. And at the time, we were looking at, there was the occasional donor who would respond to nearly everything. I think we were using a threshold of 5 and then 10%. Oh yeah, here it is, that essentially that, below 5% is green, below 10% turned yellow, and above 10 was red. So we've got this beautiful antibody with no epitopes, right? And I think, as Julia said, if you say that 10 years ago, people don't believe you. Surely the assays aren't good enough. But this one's happily going through clinical development with no immunogenicity problems. I do try and get out of my successor at Pfizer how often they get 0 epitopes. And they've got a reasonable number as far as I can tell, but it's definitely not all antibodies. So there's some challenges ahead still. I just wanted to pick up on the MAPS assay as well, so the ProPresent. Even if you've got some different sequences to play with, it's not always that you get the same kind of results purely on the sequence that you have. Also, the sequences that are presented, it's not just the context of the sequence. There'll be the other developability aspects. So another reminder that developability with regard to where the protein engineers are thinking is a good thing as well. So less sticky, less prone to aggregation. These are all good things. And so you can kind of select out, again, a parental one where we've got an H2 come up. I think this is all germline in this region, so it wasn't too much of a problem. But one of the variants, we actually then ended up with an H1 come up. So, we chose from the other variants where we weren't getting those sequence liabilities. So we had kind of the DC CD4 type setup, so the ProCern type assay as well, where you can still look at selecting or deselecting your molecules based on this cut. If you've done a good job and it's been favourable in terms of retaining potency, you can usually pick something that's low. I've done here the kind of thing that we probably shouldn't do, which is to pick some benchmark molecules and go, oh, look, it can show you in line with the ADA incidence of these molecules. But essentially, these are more like kind of hazard identification assays. So you want to ensure that you're picking from this low end. And it's usual to have some donors respond anyway, even to very low immunogenicity molecules. Okay, so just trying to put some costs on this as well. So, I mean, when you look at the costs of clinical development, they're obviously massive, but they're also really broad. You know, the range is broad, so it depends exactly what area you're working in. So it is always worth thinking about whatever we're spending in the preclinical space, there may be some relationship to, you know, what the costs are later, and indeed then potential revenues, you know, which is frankly always a guess when you're in the preclinical space. So kind of when you look through about doing the in silico and the different assays, I got some numbers from Edmund to work out, you know, the approximate cost. But even if you chose to do all of those assays, I think this is in line with what Nick was saying, you might only be spending somewhere between, you know, 1% and 4% of the IND ready package on the immunogenicity. So again, if you kind of think it's extra money, can we really afford it? You could also think if you've got one product you're taking forward, particularly if it's a platform product, you don't want to get to the clinic and then have the patients tell you that you should have spent that money. Much better to spend it ahead of time. So again, you know, if there are places where you need to spend slightly less, then you can always look at, you know, how many molecules do you really want to put through? Can you do something with fewer peptides to address the most concerning of the risks you see? Okay, so just to kind of highlight as well, when I was working within pharma, there was usually kind of mixed messages from management. Some of them were like, oh yeah, we really need to do this stuff and make sure we've got the best molecules. Others were, we're not sure we believe them, so we're going to find out in the clinic. And sometimes that meant finding the bad stuff in the clinic. And it's definitely not a good place to find out the bad stuff. But there's some common needs when you kind of think about stakeholders. So I'd always encourage you to talk to your stakeholders to see what they need and try and explain to them what you can deliver. But a risk assessment, this is a common need. Everyone needs this. If you've got investors you're working with, if you're looking to partner with a industrial company, if you're in a larger company and you've got a governance group, and then ultimately the regulators, they need to see your risk assessment. So the more detailed that is, usually the better, because it allows, well, it shows that you've done a good job, thorough job assessing your risk. It also gives you the opportunity to address those risks ahead of time. This is tricky, right? Risk of inducing impactful ADA. So generally we would say more ADA gives you a higher chance of getting more impactful ADA. So showing that you've demonstrated a lower immunogenicity strategy is a positive thing. One of the things as well is what's the possibility to mitigate, so be that through different dosing. I mean, in the early phases, your mitigation may well be, I'm running the proimmune assays to make sure that we can choose the low immunogenicity molecule. And then there's kind of specific needs people might want to know, is like, you know, the time to the potential impact. You know, if it's an investor and they want to get you into a phase 1 because they're mechanism testing and may be able to resolve things in a different way later, then that's fine. Industrial partners, they're probably looking at several different projects in the same space. How are you going to make yours stand out as something that's attractive? And certainly this is becoming an area where being able to say, I've done all the immunogenicity work, we've made a wonderful molecule, this is, you know, as good as you're going to see anywhere, is helpful. And then with, say, larger portfolio or larger companies with the portfolios, it may be a case of using it as a positioning. If there's a strategic realignment, some of the things that might fall off would be the higher immunogenicity risk. I think there's still this identification of knowing that you can detect all the potential ADA is very much where the regulatory need is. As Daniela highlighted in her talk earlier, first and foremost, it's have we measured all the ADA, or have we got the ADA assays in place? So if there are any safety outcomes, we can definitely identify whether ADA was the cause of those. So the stakeholders all have different expectations. You can use your risk assessment as a way to have those conversations in a very science-driven, pragmatic and logical way, as well as, offsetting any potential panic that will come if they only see the immunogenicity later. So with that, I want to finish up by saying the immunogenicity risk assessment does help develop successful medicines. It drives changes, and this is your route to be able to get to a medicine where later in your career you can say, I made that, and I made some good choices along the way. So we know that with the risk factors that whilst they impact the projects, we kind of understand most of them. I think even in projects that fail, we don't really look at them now and go, oh, that was a total surprise. There's always something there that's rational in terms of how we understand the immune system to work. So use these tools, the in silico and the in vitro assessments. And this final point as well, there's always a business element. However much we would love to just do the science, I think Daniela was talking about this as well, where from an agency perspective, it's always about, the patient safety, the science. For those of us who are developing the drugs, it's always about the cost of the business as well. And you have to be able to position it as a positive cost and investment that will lead to a success and not cause you additional costs further down the line. So with that, I'd like to say thanks once again to ProImmune for inviting me to come and speak to you. And thanks for all your attention. I don't know if we have time for questions, but thank you very much.
All right, thank you very much indeed, team. Another round of applause for the great presentation. So we have come to the end of this meeting, and it has been a pleasure for us to actually host you in this meeting in Shanghai. And I hope that you all have found these 2 days a fruitful journey, and that you all have absorbed lots of new information, new knowledges in terms of how you can actually integrate and strategize the way that how you can actually develop your biotherapeutics program in your company. So in terms of the meetings or the content, so they are all recorded, and so what we'll be doing next is that we will be starting to edit them, and then we will make all this content to be available for free onto the public domain onto YouTube. But I do know that there might be some challenges in China, so it will be also be available on B-Zhan, or we call it Bilibili. Yeah, that's where we have been actually uploading a lot of these academia contents and talks from our past meetings. So feel free to look out for our channel, and you can actually find a lot of talks that I think 15 years ago, 11 years ago, Tim actually attended our meeting and also gave a talk regarding immunogenicity at that point of time. Yeah, and I would like to welcome CEO of ProImmune to give a final speech before we close this meeting. Please.
Yeah, thank you, Edmund, and thank you again for coming. It won't be a long speech, but yeah, I mean, we've pulled this conference together, sort of, I wouldn't say last minute, but with a very pressured time frame. So I want to extend my thanks to my team who've worked incredibly hard to make this happen. And also I was amazed by the support that we've gotten here from the consulate in Shanghai, also the development organization here, and frankly also the team of suppliers here on the ground helping with AV and everything. Setting everything up, including the team here at the Oriental Riverside Hotel and the Convention Center, which was amazing. And I think you would probably agree that this is an amazing venue to have this conference. Yeah, so I hope you got some interesting information out of this meeting. We've been thinking that there is a gap, I guess, in terms of an opportunity to network between people like Daniela, the FDA, etc. For them to come here to present their perspective on drug development and immunogenicity in the United States, for us to give a sort of overview of some of the things that we've been doing. I know the biopharmaceutical industry in China is changing very rapidly. We are still trying to understand it. I mean, you're probably also still trying to sort of understand what exactly is the shape of all of your opportunities, you know, bringing drugs to the West, to countries like the US and Europe. And of course, there's a huge, enormous growing market in China. There are slightly different probably priorities in the two different markets in terms of, you know, where the medical need is. I think that's also a very interesting sort of question to think about in the future. There'll be a lot of Western companies that want to market their drugs in China. Then there's the entire investment sort of life cycle. I talked a little bit about the finance, but ultimately, it's always a question of where is all that money coming from that's supporting drug development. The US is by far the most established market in that, with obviously a very well-established industry of private equity, venture capitalists, and also large pharma companies spending their money to fund new companies and drug development. And there's a sort of relatively well-established process in that. I have a sense that in China, it's still a little bit sort of in a stage of maturing. So be interesting to see how that works. It's for everyone involved. It's important to understand where is the return on investment. That's also the reason of my talk from yesterday. Ultimately, developing drugs is very expensive. It's very high risk as well, let's face it, compared to other propositions. And so, you know, in science, it's important to, understand what's going on. You can't predict everything, so it's a sort of risk-return analysis, and you need to understand the risk. Hopefully, we have been able to give you some more perspectives on that, how you can do it. I think the last thing I want to say is that, you know, in addition to providing tools and services to people, we also want to, and we see ourselves as, you know, trying to bring people together, right? To create opportunities for people to meet, to talk to each other. And that can be us, of course. It can be making connections with some of the speakers that you've heard. I don't know if before this meeting you felt that the FDA was very approachable, but they are. You know, as Zubin said, if you have any questions, you can just email him. And if it's a scientific question, rather than the question, will you approve my drug, then he will answer. So the last thought I want to leave you with is that, you know, if you want to make any connections, any contacts with anybody about anything, then talk to Edmund Wenzel, get in touch with us. We will put you directly in contact with the relevant people. We've been in this game, well, you know, general immunology since the start of our company a quarter century ago, seems like yesterday, but in immunogenicity for over 20 years. So we've done a lot of things with a lot of people, and we can help you connect to anyone you want to get in touch with. And so with that, thank you everyone for coming, and hope you attend next time. Thank you.
Maybe they got a teaser of